Imagine a book that holds the instructions for building and maintaining a living organism. This book is DNA, the blueprint of life. It contains all the genetic information that makes you, you. Each cell in your body holds a complete copy of this book, written in a language of chemical letters called bases. Just like a book with chapters and sentences, DNA is organized into genes. Genes are specific segments of DNA that carry the instructions for building proteins, the building blocks of life. They determine our traits, from eye color to susceptibility to certain diseases. Changes in the DNA sequence, like typos in a book, can lead to errors in protein production and potentially cause diseases. This is where CRISPR comes in, offering a revolutionary tool to edit these errors and rewrite the code of life. CRISPR, short for clustered regularly interspaced short palindromic repeats, wasn't initially designed for editing human genes. Its origins lie within the humble bacterial cell, acting as an adaptive immune system against invading viruses. Think of it as a bacterial most wanted poster system. When a virus attacks a bacterium, it injects its DNA into the cell. The CRISPR system captures snippets of this viral DNA and inserts them into the bacterium's own genome, creating a memory bank of past infections. These stored viral DNA fragments act as mugshots of the virus. If the same virus attacks again, the bacterium can quickly recognize and destroy the viral DNA using the information stored in its CRISPR system. This remarkable defense mechanism sparked the interest of scientists, leading to its adaptation as a powerful gene editing tool. CRISPR's gene editing capability relies on two key components, a guide RNA molecule and a Cas9 protein. The guide RNA acts like a GPS system designed to locate a specific target sequence in the DNA. It carries a short sequence that matches the DNA sequence of the gene we want to edit. The Cas9 protein acts like a pair of molecular scissors. Once the guide RNA locates the target DNA sequence, the Cas9 protein binds to it and makes a precise cut, much like a surgeon making a precise incision. This cut disables the targeted gene, preventing it from producing its protein. After the cut, the cell's natural DNA repair mechanisms kick in. These mechanisms can be harnessed to introduce changes to the DNA sequence. We can either disrupt the gene entirely or even insert a new piece of DNA, effectively rewriting the genetic code at the target location. The potential of CRISPR in medicine is vast, offering hope for treating and even curing genetic diseases that were once considered incurable. Imagine a future where we can correct genetic defects that cause cystic fibrosis, sickle cell anemia, or Huntington's disease. CRISPR's precision in targeting and editing specific genes makes it a powerful tool for developing personalized therapies. Researchers are exploring its use in creating CAR T-cell therapies for cancer, where a patient's immune cells are engineered to target and destroy cancer cells more effectively. Beyond treating diseases, CRISPR holds promise for preventing them altogether. By correcting genetic mutations in embryos, we could potentially eliminate inherited diseases from future generations. However, this possibility also raises significant ethical concerns that require careful consideration. Section 5. Agricultural advancements with CRISPR. CRISPR's impact extends beyond human health, revolutionizing agriculture and food production. By editing the genes of crops, scientists can enhance desirable traits like yield, nutritional content, and resistance to pests and diseases. Imagine fields of drought-resistant crops thriving in arid regions, reducing food shortages and improving livelihoods. CRISPR can create crops that require fewer pesticides and herbicides, benefiting the environment and human health. The ability to tailor crops to specific environments and nutritional needs has the potential to transform global food security. We can develop crops enriched with essential vitamins and minerals, addressing malnutrition in vulnerable populations. However, ethical and regulatory considerations surrounding genetically modified organisms, or GMOs, remain crucial. Section 6. Ethical considerations, a double-edged sword. While CRISPR holds immense promise, its unprecedented power to alter the building blocks of life raises profound ethical considerations. The ability to edit the human germline, meaning changes that would be passed down to future generations, is particularly controversial. Concerns arise about the potential for unintended consequences of germline editing. What if edits inadvertently introduce new mutations or create unforeseen health problems in future generations? The long-term effects of such modifications are still unknown and require extensive research. Furthermore, the possibility of designer babies, children with genetically selected traits, raises ethical dilemmas about human enhancement and genetic inequality. Who gets to decide which traits are desirable and who has access to such technologies? These are complex questions with no easy answers. Section 7. The future of CRISPR, limitless possibilities. Despite the ethical complexities, the future of CRISPR is brimming with possibilities. As our understanding of gene editing deepens and technology advances, we can anticipate even more groundbreaking applications in various fields. Imagine a world where genetic diseases are eradicated, where organ transplantation is replaced by gene-edited organs grown in labs, and where aging itself is potentially reversible. CRISPR could revolutionize biofuel production, create new materials with enhanced properties, and even contribute to conservation efforts by protecting endangered species. The potential benefits of CRISPR are vast, offering solutions to some of humanity's most pressing challenges. However, realizing this potential requires responsible development, ethical oversight, and open public dialogue to navigate the complex implications of this revolutionary technology. Section 8. A world transformed. The promise and peril of CRISPR. CRISPR has ushered in a new era of genetic engineering, offering unprecedented power to rewrite the code of life. From curing diseases to enhancing crops, its potential applications are vast and transformative. However, this power comes with profound ethical considerations that we must carefully address. As we stand on the cusp of this genetic revolution, we must ask ourselves, how do we harness the potential of CRISPR while mitigating its risks? How do we ensure equitable access to these transformative technologies? The future of life itself may depend on our ability to answer these questions thoughtfully and responsibly. The journey into the world of gene editing has just begun, and the choices we make today will shape the future of life for generations to come.